All right, ready to dive in. Today, we're going to tackle some familiar foes, especially for you medical microbiologists out there.
Oh, yeah.
E. coli and Shigella species.
Sounds fun.
And we're using Principles and Practice of Clinical Bacteriology, second edition, by Gillespie and Hockey as a roadmap. So get ready to, refresh your knowledge, maybe even pick up some new insights along the way.
Yeah, it's amazing how these two organisms can be so closely related, but have such different impacts on human health. E. coli, a common resident in our gut, and Shigella, a notorious bad guy, causing a lot of illness.
Yeah, exactly. And that's what makes this deep dive so interesting, right?
Yeah.
Starting with E. coli. I mean, it's been a mainstay in microbiology labs since Dr. Theodore Escherichia first identified it way back in 1885.
Wow.
And who knew it would become such a superstar, especially that K12 strain?
Right. I mean, E. coli K12 has been instrumental in uncovering so much of what we know about bacterial genetics, but it's crucial to remember that E. coli is incredibly diverse. You have your harmless commensals, right? And then you have the strains packing some serious pathogenic potential.
Let's unpack that diversity a bit. We know the basics, right? Gram-negative rods, often motile, facultative anaerobes, and usually ferment lactose. But how does that translate to the different ways that they can actually cause disease?
Well, we can categorize pathogenic E. coli into two main groups, really: extraintestinal pathogenic E. coli, or XPEC, and then you have intestinal pathogenic E. coli, or IPEC. It's like they've specialized in causing trouble in different parts of the body.
And to make things even more complicated, within those groups, there are further classifications based on their specific virulence factors and the types of infections they cause.
Absolutely, yeah. The evolution of these different pathotypes is a fascinating story of adaptation and survival, largely driven by horizontal gene transfer. It's almost like bacteria swapping genetic material, like trading cards, picking up new tools to survive and thrive.
So these bacteria are constantly evolving, picking up new tricks to outsmart our immune systems and cause disease.
Precisely, yeah. And one of the key players in this evolutionary arms race is something called pathogenicity islands, or PAIs. These are clusters of genes that code for virulence factors, often acquired through those genetic swaps we talked about.
It's like they're installing upgrade packages, adding new weapons to their arsenal.
That's a great analogy. Yeah, and when you compare the genome size of a harmless E. coli strain like K12 to that of a highly pathogenic strain like O157.H7, the difference in these PAIs is striking. It's almost like comparing a basic toolkit to a high-tech weapons lab.
Okay, so we've got these different groups of pathogenic E. coli with varying levels of weaponry. Let's talk about what that actually means for us in terms of clinical features. What kinds of infections do they cause?
So let's start with XPEC. As the name suggests, these are the strains that cause infections outside the gut. And they can be quite versatile, causing UTIs, bloodstream infections, even meningitis, especially in newborns.
And for you medical microbiologists out there, just think about those classic UTI symptoms. The urgency, the burning, the frequent trips to the bathroom, and most of the time it's E. coli to blame.
And those UTIs can sometimes escalate to pyelonephritis, which is an infection of the kidneys. And that's when things get more serious, fever, chills, flank pain, and even sepsis in some cases.
Not to mention the potential for E. coli to enter the bloodstream, causing bacteremia. And as we mentioned, those specific strains with the K1 capsule, particularly dangerous for newborns, leading to meningitis.
That K1 capsule, it's a real game changer. It helps the bacteria evade the immune system and survive in the bloodstream, which makes those infections much harder to treat.
Speaking of challenging infections, let's move on to the intestinal pathogenic E. coli, or IPEC. This is where we encounter those 6 major pathotypes, each with its unique set of virulence factors and clinical presentations.
Right. We have enteroinvasive E. coli, or EIEC, enterotoxigenic E. coli, or ETEC, enteropathogenic E. coli, EPEC, enterohemorrhagic E. coli, EAC, interaggregative E. coli, and diffusely adherent E. coli, or DAEC. It's a mouthful, I know. But each of these groups is responsible for a distinct type of diarrheal illness.
I can think of them like a rogues gallery of gut disruptors, each with its own modus operandi.
Exactly. And it's important to understand how they differ in their mechanisms of pathogenicity so you can accurately diagnose and treat these infections.
So let's start with EIC. These guys are fascinating because they act almost like Shigella, invading the cells of the intestinal lining.
It's true. They even share some biochemical similarities with Shigella, often being non-motile and lysine decarboxylase negative. And just like Shigella, they can cause bacillary dysentery, a severe form of diarrhea with blood and mucus.
So we've got these invasive E. coli causing direct damage to the gut lining. But then we have other pathotypes, like ETEC, that take a different approach.
Right. ETEC are known for producing toxins that disrupt fluid balance in the intestines, leading to watery diarrhea. It's the classic traveler's diarrhea scenario.
So instead of invading cells, they're waging a chemical warfare campaign, disrupting the gut's normal function from afar.
Very apt description, yeah. And then you have EPEC, which are masters of attachment. They adhere to the intestinal cells and cause these characteristic attaching and effacing lesions, disrupting the structure of the gut lining.
It's like they're remodeling the intestinal landscape to their advantage, causing inflammation and malabsorption.
Precisely. And let's not forget about EHE, the notorious group that includes O157 or H7, a major cause of those foodborne outbreaks. They produce those potent Shigella-like toxins, which can lead to bloody diarrhea and even hemolytic uremic syndrome, or HUS.
And HUS is a serious complication, potentially causing kidney failure.
Yeah.
It's a stark reminder of how devastating these E. coli infections can be.
And then we have the lesser known, but it's still significant, EHE and DAEC. They're associated with persistent diarrhea, especially in children. AG forms these characteristic aggregates on the intestinal lining, while DAEC adheres diffusely, both disrupting the normal gut function.
So it's like they're playing different roles in this intestinal drama, each contributing to the overall disruption and disease. But with so much diversity, how do we go about differentiating these various E. coli pathotypes in the lab?
Well, it's a combination of traditional methods and more modern techniques. Classic tests like lactose fermentation on McConkey agar, growth at 44 degrees Celsius, and indole production can help narrow down the possibilities.
But those classic tests aren't always foolproof, are they?
Exactly. Some pathogenic strains, like EIEC, can be slow lactose fermenters or even non-lactose fermenters, mimicking Shigella in that regard. And other bacteria can share similar characteristics with E. coli, leading to false positives. It's a bit of a detective game.
So what are the tools that have really revolutionized E. coli identification?
Well, chromogenic media have been a real game changer. They exploit specific enzyme activities within the bacteria to produce colonies of distinct colors, making identification much easier. For example, many target the beta-glucuronidase enzyme, which most E. coli possess, but interestingly not O157.H7.
It's always good to have a few tricks up your sleeve when dealing with these bacteria. They seem to enjoy keeping us on our toes.
They certainly do, yeah. And then we have the power of molecular techniques like DNA probes and PCR, which can target specific genes within the bacteria, allowing us to quickly and accurately identify different pathotypes.
It's amazing how technology has advanced our diagnostic capabilities. It's like having a microbial fingerprint scanner right there in the lab.
Absolutely. And these tools are essential not only for identifying the specific E. coli pathotype, but also for tracking the spread of antibiotic resistance, which is a growing concern.
Speaking of antibiotic resistance, how are we faring in the battle against these increasingly resistant E. coli strains?
Unfortunately, it's an ongoing challenge. The widespread use of antibiotics has driven the selection and spread of resistance genes, often carried on plasmids or other mobile genetic elements. We're seeing decreasing susceptibility to many commonly used antibiotics, such as amoxicillin, trimethoprim, sulfamethoxidil, and even fluoroquinolones in some cases.
So it's like a microbial arms race with the bacteria constantly evolving new defenses against our antibiotics.
That's a great way to put it, yeah. But there is some good news. Some antibiotics like nitrofurantoin and fosfomycin remain highly effective against most urinary tract infections caused by E. coli. And for more serious infections, we have carbapenems, although we need to use them judiciously to preserve their effectiveness.
It's a delicate balancing act, isn't it? We need to treat these infections effectively, but also be mindful of the long-term consequences of antibiotic overuse.
Absolutely. Antimicrobial stewardship is crucial. It's about using antibiotics wisely only when necessary, and choosing the right antibiotic for the right bug based on susceptibility testing.
So it's about personalized medicine for bacteria.
Exactly.
But before we delve further into the treatment and management of E. coli infections, let's shift our focus to Shigella, a closely related organism, but one that causes a very distinct clinical syndrome.
Indeed. Shigella is a true master of stealth and disruption when it comes to the human gut.
So let's break down the basics. What makes Shigella unique compared to E. coli?
Well, like E. coli, they're gram-negative rods. But unlike most E. coli, Shigella species are non-motile, and they ferment glucose without producing gas. And they're even more specialized in their niche, targeting the human colon almost exclusively.
So they're like the elite special forces unit of the bacterial world, highly trained for a specific mission.
A very fitting analogy, yeah. And their mission is to invade the cells lining the colon, causing intense inflammation and leading to those classic symptoms of dysentery.
And dysentery has a long and rather unpleasant history in human health, hasn't it?
Absolutely, yeah. It's been responsible for countless outbreaks and epidemics throughout history, often associated with poor sanitation and crowded living conditions. And even today, it remains a major public health concern in many parts of the world.
So how does Shigella pull off this invasion? It sounds like a well-coordinated attack.
It is, yeah. They first breach the intestinal barrier, often by exploiting specialized cells called M cells, which are meant to sample antigens from the gut lumen.
So they sneak in through the back door, so to speak.
Exactly. Once inside, they launch a full-on assault, killing macrophages, releasing cytokines, and attracting a flood of immune cells, creating chaos in the gut.
It's like they're triggering a biological alarm, but they're the ones setting off the fire.
Precisely. And as the immune response intensifies, it actually helps them break down the intestinal barrier even further, allowing them to spread and invade more cells.
Talk about a clever, albeit destructive, strategy.
And it doesn't stop there. They have this special protein called icosay that allows them to hijack the host cell's own machinery, essentially creating an actin tail that propels them from one cell to the next.
So they're not just invading, they're spreading like wildfire through the intestinal lining.
Exactly. And to make matters worse, some Shigella species, particularly Shigella dysenteriae type 1, produce a potent toxin called Shiga toxin or Shiga tox.
And we talked about Shiga-like toxins produced by certain E. coli strains. Are they similar?
They're very similar, yeah. They both target ribosomes in host cells, disrupting protein synthesis and leading to cell death. This contributes to the severe damage seen in dysentery and can also have systemic effects, potentially leading to HUS.
So we have this combination of direct invasion, inflammation, and toxin production, all contributing to the debilitating symptoms of dysentery. It's no wonder this infection can be so severe, especially in young children and those with compromised immune systems.
That's right. But let's not get too bogged down in the doom and gloom. We do have tools to fight back, both in terms of diagnosis and treatment.
That's what I like to hear. So how do we go about identifying Shigella in the lab? Are the methods similar to those used for E. coli?
There are some similarities, but also some key differences. Like E. coli, they're non-lactose fermenters, so they'll appear as colorless colonies on McConkey agar. However, Shigella is a bit more fastidious, and it doesn't survive well outside the host.
So prompt processing of clinical specimens is crucial, right?
Absolutely. And because they share some biochemical similarities with other Enterobacteriaceae, we rely on a combination of selective media, biochemical tests, and serological typing to confirm the diagnosis.
And I imagine molecular techniques like PCR are playing an increasingly important role in Shigella identification as well.
Definitely, yeah. PCR, targeting the IPH gene, which is involved in Shigella's invasive capabilities, is a rapid and sensitive diagnostic tool. It's become an invaluable tool in outbreak investigations and epidemiological studies.
It sounds like we have a pretty good handle on identifying Shigella. But what about treatment? Do antibiotics play a role in managing these infections?
In many cases, shigellosis is self-limiting, and supportive care with oral rehydration therapy is sufficient. However, antibiotics can shorten the duration of illness, reduce the severity of symptoms, and limit the spread of the bacteria.
But antibiotic resistance is a concern here as well, right?
Unfortunately, yes. We're seeing increasing resistance to commonly used antibiotics like ampicillin and trimethoprim sulfamethoxazole.
So what are our options when those first-line antibiotics fail?
Fluoroquinolones, like ciprofloxacin, are often effective, but resistance is emerging. In severe cases or when fluoroquinolone resistance is suspected, third-generation cephalosporins or azithromycin may be used.
It sounds like a constantly evolving challenge with both E. coli and Shigella, finding new ways to outsmart our antibiotics.
It certainly is. That's why antimicrobial stewardship and the development of new therapeutic strategies are so crucial in the fight against these pathogens.
Well said. It's a reminder that we can't become complacent. We need to stay vigilant, stay informed, and keep pushing the boundaries of research and innovation.
Absolutely.
So on that note, we've covered a lot of ground today. We've explored the intricate world of E. coli and Shigella, from their basic characteristics and virulence factors to their clinical presentations, epidemiology, and treatment strategies.
And we've delved into the challenges of antibiotic resistance and explored some of the exciting new approaches that hold promise for the future.
It's been a real deep dive, and hopefully our listeners have gained some valuable insights to take back to their labs and clinics.
Remember, knowledge is our most powerful weapon in this ongoing battle against these microscopic foes. Stay informed, stay curious, and stay vigilant.
All right, so before we let you go, I want to leave you with something to ponder. We've talked about the challenges we're facing, but what about the future? What emerging technologies or approaches are giving you hope in this fight against increasingly resistant bugs like E. coli and Shigella?
That's a great question, and one that keeps researchers busy around the clock, really. You know, we touched on phage therapy earlier, and it's definitely showing promise for a variety of bacterial infections. But there are other exciting avenues being explored as well.
Do tell. What's on the horizon that has you particularly intrigued?
Well, one area that I find fascinating is the development of novel antibiotics that target bacterial virulence factors.
Yeah.
Instead of those essential cellular processes that we usually go after, right? The idea is to disarm the bacteria without pushing them to evolve resistance quite so aggressively.
So instead of trying to kill the bacteria outright, we're finding ways to take away their weapons.
Exactly. And if we can disrupt their ability to cause disease without putting that same selective pressure on them, it could be a real game changer in slowing down resistance development.
That's a brilliant strategy. Are there other approaches that have that same disarming philosophy?
There are. Researchers are exploring anti-virulence therapies that target things like toxin production, adhesion to host cells, and even those communication systems bacteria use to coordinate their attack.
Wow, it's like we're learning to speak their language so we can disrupt their strategies.
In a sense, yes. And then there's the whole field of nanotechnology, which is opening up incredible possibilities for targeted drug delivery. Imagine delivering antibiotics directly to the site of infection using nanoparticles. Increased efficacy, fewer side effects. It's a win-win.
Nanoparticles delivering antibiotics directly to the battlefield. That does sound like something straight out of science fiction.
Yeah.
What other futuristic tools are we adding to the arsenal?
Well, we can't forget about the microbiome. We're only beginning to grasp the intricate relationship between our gut microbes and our health, but the potential for therapeutic manipulation is huge.
So we're talking about harnessing the power of our own gut bacteria to fight off these infections.
Mm-hmm.
It's like recruiting our internal army of microbes to defend against invaders.
That's the idea. It's still early days, but research is showing that manipulating the composition and function of the gut microbiome can have a real impact on our susceptibility to infections, including those caused by E. coli and Shigella.
It's mind-blowing to think that the future of medicine might lie within our own bodies, within those trillions of microbes we often take for granted.
It truly is a paradigm shift in how we think about health and disease, and it highlights the importance of interdisciplinary collaboration. We need microbiologists, immunologists, geneticists, and clinicians all working together to unravel these complexities and develop innovative solutions.
It's a team effort, and a global one at that. We're all connected in this microbial world, and sharing knowledge and resources is crucial to staying ahead of these evolving threats.
Couldn't agree more. Collaboration is key to tackling these complex challenges and ensuring a healthier future for all.
Well, on that inspiring note, we'll wrap up this deep dive into the world of E. coli and Shigella. We hope you found it insightful, engaging, and maybe even spark some new ideas for your own work.
And remember, as medical microbiologists, you're on the front lines of this battle. Your expertise and dedication are essential to protecting public health and tackling these ever-evolving microbial challenges.
To all our listeners out there, keep exploring, keep learning, and keep those lab coats spotless. Until next time, stay curious.
All right, question number 1. Which of the following is not a characteristic feature of Escherichia coli? Is it A, gram-negative rod, B, facultative anaerobe, C, oxidase positive, or D, often motile by paratricious flagella?
Think back to those fundamental characteristics we discussed earlier. What sets E. coli apart from other bacteria?
The correct answer is C, oxidase positive. E. coli is actually oxidase negative, and the simple test can be a useful tool in differentiating it from other enterobacteriaceae.
It's those seemingly small details that can make a big difference in the lab. Okay, on to question number 2. Pathogenicity islands, or PAIs, play a significant role in the virulence of various E. coli pathotypes. What are PAIs?
Are they A, small circular DNA molecules that can replicate independently, B, clusters of virulence-associated genes that are often located on mobile genetic elements, C, specific regions on the bacterial chromosome where genes for antibiotic resistance are found, or D, structures on the bacterial surface that help them adhere to host cells?
This one really gets to the heart of how E. coli evolves and becomes pathogenic. What's the key takeaway here for our listeners?
The answer is B, clusters of virulence-associated genes that are often located on mobile genetic elements. These PAIs are like genetic hotspots for virulence factors acquired through horizontal gene transfer. They're a major driving force in the emergence of new and often more dangerous E. coli strains.
It's like they're constantly upgrading their software, adding new features to enhance their ability to cause disease. Okay, question 3. Which E. coli pathotype is most commonly associated with urinary tract infections or UTIs? Is it A, enterotoxigenic E. coli or ETEC, B, uropathogenic E. coli or UPEC, C, enterohemorrhagic E. coli or EHEC, or D, enteroinvasive E. coli or EIEC?
Consider the specific environments these bacteria inhabit and the types of infections they cause.
And remember, we're talking about those pesky UTIs that bring so many patients to the clinic.
The correct answer is B, uropathogenic E. coli or UPEC. These strains are equipped with a unique arsenal of virulence factors that allow them to colonize the urinary tract and cause those all too familiar symptoms.
They're like the ultimate bladder invaders. Now let's shift gears slightly and talk about toxins. Question 4. Shigella toxin, produced by Shigella dysenteria type 1, is known to A, increase intestinal motility, leading to watery diarrhea, B, inhibit protein synthesis in host cells, causing cell death, C, stimulate the production of cyclic AMP, resulting in fluid secretion, or D, bind to guanylate cyclase receptors, leading to electrolyte imbalance.
Think about the devastating effects of Shigella toxin both locally in the gut and systemically. It's a potent weapon in Shigella's arsenal.
It's not messing around. What does it do at the cellular level?
The correct answer is B, inhibit protein synthesis in host cells, causing cell death. Shigella toxin acts like a molecular saboteur, disrupting this essential process and leading to widespread cell death. This contributes to the severe damage seen in dysentery and can also have devastating effects on other organs like the kidneys and HUS.
It's a powerful reminder of the destructive capabilities of these bacterial toxins.
Yeah.
Okay, last question, and this one is a bit trickier. The use of antibiotics in the treatment of E. coli 0157-borne H7 infections is controversial because A, E. coli 0157-borne H7 is inherently resistant to most antibiotics. B, antibiotics can induce the production and release of Shigella-like toxins, potentially worsening the disease. C, antibiotics have no effect on the symptoms of E. coli 0157-borne H7 infection. Or D, antibiotics can prolong the duration of the illness.
Think about the unique characteristics of this particular strain and its relationship with Shigella-like toxins.
This gets to the heart of those challenging clinical decisions we face as medical microbiologists. It's not always a straightforward answer.
Exactly. The correct answer is B, antibiotics can induce the production and release of Shigella-like toxins, potentially worsening the disease. While antibiotics are our go-to weapon for many bacterial infections, in the case of E. coli 0157.H7, they can actually backfire, triggering the release of even more toxins and increasing the risk of complications like HUS.
It's a crucial point for any medical microbiologist to remember.
Yeah.
Sometimes less is more when it comes to treatment. All right, listeners, how did you do on our pop quiz? Hopefully you aced it, but even if you missed a few, it's a great opportunity to revisit those key concepts.
Remember, lifelong learning is an essential part of our profession. The world of bacteria is constantly evolving, and we need to stay informed and adaptable to provide the best possible care for our patients.
Well said. Now, shifting gears a bit, let's zoom out and look at the bigger picture of epidemiology. It's like detective work on a grand scale, trying to understand how these infections spread and impact populations.
Exactly. Epidemiology helps us identify risk factors, track outbreaks, and ultimately develop strategies to prevent and control these diseases.
So when it comes to E. coli and Shigella, what are some of the key epidemiological insights?
Well, E. coli is ubiquitous. We've talked about how diverse it is, and that diversity extends to its habitats as well. We find E. coli in the guts of humans and animals, in the environment, practically everywhere. But the pathogenic strains, particularly those causing diarrheal illness, are often associated with specific sources like contaminated food and water.
Think undercooked meat, unpasteurized milk, contaminated produce, those classic culprits we always warn our patients about.
Precisely. And those foodborne outbreaks can be particularly challenging to control, especially when they involve those widespread distribution networks.
It's a reminder that food safety is a crucial aspect of public health.
Absolutely. Now, Shigella is a bit different. It's primarily a human pathogen, and its transmission is closely linked to poor sanitation and hygiene.
So we're talking about crowded living conditions, lack of access to clean water and sanitation facilities, those kinds of environments.
Exactly. And that's why Shigella infections are a major problem in developing countries, especially among young children. The WHO estimates millions of cases and over 1 million deaths annually due to Shigella. It's a stark reminder of the global health disparities that still exist.
It's heartbreaking, but it also highlights the importance of public health interventions like improving sanitation, promoting hand hygiene, and ensuring access to safe water.
Those are fundamental strategies for breaking the cycle of transmission, and we can't forget about vaccines. Research is ongoing to develop effective vaccines against Shigella, which could be game changers in the fight against this disease.
Vaccines are like the ultimate preventive measure training our immune systems to recognize and neutralize these pathogens before they can even cause disease. Speaking of prevention, let's circle back to antibiotic resistance for a moment.
Yeah.
It's a major threat to our ability to effectively treat both E. coli and Shigella infections.
Mm-hmm.
What can we do to combat this growing problem?
Well, it's a multi-pronged approach. We need to promote responsible antibiotic use both in healthcare settings and in the community. That means reducing unnecessary prescriptions, ensuring antibiotics are used only for bacterial infections, and using the right antibiotic for the right bug based on susceptibility testing.
Education is key, isn't it? We need to raise awareness among healthcare providers and the public about the importance of antimicrobial stewardship.
Absolutely. It's about using antibiotics wisely to preserve their effectiveness for future generations.
It's like we're stewards of these precious resources, and we need to use them responsibly.
Exactly. And beyond stewardship, we need to invest in research and development of new antibiotics, as well as alternative therapies like phage therapy and immune-based approaches.
Phage therapy, that sounds intriguing. Can you tell us more about that?
It's a fascinating approach that utilizes viruses called bacteriophages that infect and kill bacteria. It's like using nature's own weapons to fight bacterial infections.
Yeah, we're enlisting these tiny viral assassins to target the bad bacteria.
Precisely. And because phages are highly specific for their target bacteria, they offer the potential for very targeted therapy with minimal disruption to the patient's normal microbiome.
That's a huge advantage over broad spectrum antibiotics, which can often wipe out both good and bad bacteria, leading to unintended consequences.
Exactly. And there's also growing interest in manipulating the gut microbiome to enhance resistance to pathogens like E. coli and Shigella.
So instead of just targeting the pathogens directly, we're trying to modulate the entire ecosystem of the gut to make it less hospitable to them.
That's the idea. It's a more holistic approach that recognizes the complex interplay between our gut bacteria and our overall health.
It's fascinating to think that the key to fighting these infections might lie within our own bodies, within the trillions of microbes that inhabit our gut.
It's a reminder that we're not just individuals, but complex ecosystems, and our health is intricately linked to the health of our microbiome.
Well said. It sounds like there's a lot of A lot of promising research on the horizon, offering hope for new ways to combat these ever-evolving bacterial threats.
Definitely. But for now, it's back to the basics, promoting good hygiene, practicing antimicrobial stewardship, and staying informed about the latest developments in this fascinating and challenging field.
Well, on that note, we've covered a lot of ground today. We've explored the intricate world of E. coli and Shigella, from their basic characteristics and virulence factors to their clinical presentations, epidemiology, and treatment strategies.
And we've delved into the challenges of antibiotic resistance and explored some of the exciting new approaches that hold promise for the future.
It's been a real deep dive, and hopefully our listeners have gained some valuable insights to take back to their labs and clinics.
Remember, knowledge is our most powerful weapon in this ongoing battle against these microscopic foes. Stay informed, stay curious, and stay vigilant.
So it truly is a paradigm shift in how we think about health and disease.
Yeah.
And it highlights the importance of interdisciplinary collaboration.
Oh, absolutely.
Well, we need microbiologists, immunologists, geneticists, and clinicians all working together to unravel these complexities and develop innovative solutions.
It's a team effort, and a global one at that. We're all connected in this microbial world, and sharing knowledge and resources is crucial to staying ahead of these evolving threats.
Couldn't agree more. Collaboration is key to tackling these complex challenges and ensuring a healthier future for all.
Yeah.
Well, on that inspiring note, we'll wrap up this deep dive into the world of E. coli and Shigella. We hope you found it insightful, engaging, and maybe even spark some new ideas for your own work.
And remember, as medical microbiologists, you're on the front lines of this battle. Your expertise and dedication are essential to protecting public health and tackling these ever-evolving microbial challenges.
To all our listeners out there, keep exploring, keep learning, and keep those lab coats spotless.
Right.
Until next time, stay curious.